Well, I'd like to welcome Dr. Samir Sonkusale to the Resilient Biome Podcast. It's a pleasure to meet you, and thanks for joining us today.
Thank you for having me, Richard.
Yeah, I'm really excited about this topic, the ingestible microbiome sampling pill. This is going to be interesting. I'd like to read your bio, not your whole bio. That might take a long time with all your accomplishments, but just to give our listeners an idea of where you're coming from. Samir Sonkusale is currently professor of electrical and computer engineering with a joint appointment in the Department of Biomedical Engineering and also the Department of Chemical and Biological Engineering at Tufts University. He directs an interdisciplinary research group, Nano Lab, with research focus on bioelectronics, biomedical devices, circuits, and systems, nanoscale sensors, lab-on-chip microsystems, point-of-care diagnostics, analog and mixed signal computing, and brain-inspired machine learning. Dr. Sonkusale is a prolific inventor of several new devices and concepts, including smart bandages, smart sutures, and lab on a pill. His innovation on lab on a pill was listed as one of the top 8 biomedical innovations in the world for the year 2020 in the Stat Madness Challenge. The technologies developed in his lab have been licensed to companies and have resulted in the creation of several startups. So I know you're very busy. You just got back from Brazil. So again, I appreciate your time here. One of the things that stands out to me in your bio is that you're a professor of electrical and chemical engineering, but you're also working in the biomedical engineering and Department of Chemical and Biological Engineering. So wondering how you took your initial primary disciplines and moved into these interdisciplinary research.
So first of all, thanks, Richard, for having me here in this podcast. When I started my career, I mean, I've been trained as an electrical engineer. So when I started my career as an academics, I really wanted to explore how my background and my expertise in engineering can be used or utilized to address some of the global challenges. It is easier to work within the confines of disciplines, but I realize that most of the challenges are actually at the boundaries of disciplines. They are not actually within the discipline itself. So it needed, and I enjoyed that challenge. And I was, I'm an electrical engineer. I developed circuits and integrated circuits, chips that go into your phones and your microprocessors and your audio and video equipment. But then I realized that how do I make chips for sensing things? And how do I build new devices using things that I have done before? And that got me into this whole world of medicine and healthcare. There are a lot of very interesting problems in medicine and healthcare that when I was exploring this universe of what I can do with my expertise, it seemed like the best fit for me in terms of what I can bring to this discipline. And so when I started branching out, I have been in academia for many years. And curiosity, I'm driven by curiosity-driven innovation. So wherever my curiosity took me and wherever the problems are, I would basically explore that till I am able to find a solution or give up finding a solution. Both of them are fine, but and that's why you would see me doing in biomedical engineering and chemical and biological engineering. So at the moment, my lab does work on quite a lot of different interesting problems from healthcare to medicine to the environment. So I'm quite excited about that. Yeah.
Fascinating. I love how you describe it being on the edge of those disciplines because life overlaps, so therefore your research has to overlap, is what I'm hearing.
Correct. Yeah, exactly.
Yeah, absolutely fascinating. I'm sure your bio doesn't list all of your inventions and accomplishments, but which of those have gained the most traction over the years?
The one that I started working on, which is now also a startup, is related to microneedles for transdermal drug delivery, is one of the innovations that came out of the lab. and little these tiny microneedles that can penetrate the outer layer of the skin to deliver drugs. So if you ask what are the different ways we take medicines, we either have oral tablets or we take injections, right? But oral tablets, not all medicines can pass through our gastric system, and not all drugs get absorbed equally into our bloodstream effectively. So that's why we go to injections and IV drips, because they directly deliver drugs into the bloodstream. But that cannot be taken, for example, in the comforts of your home. So transdermal delivery is a way to deliver some high potency drugs directly through the skin. So it doesn't go into the bloodstream, it goes into the interstitial fluid space that is close to the blood capillaries. And it is a non-invasive, painless way for delivering drugs. It is being used now by a lot of companies for delivering vaccines, because people are afraid of taking injections. You just put a vaccine patch on. But you can deliver microRNA vaccines in the future, or all kinds of different drugs. It can also be used to deliver all kinds of other compounds into the bloodstream that you wouldn't be possible to take with a pill or an injection. So that, I would say, found the most traction in terms of all the way from innovation in the lab to actually having a company out there. But I'm also equally proud of the microbiome sampling pills. That is actually probably one of my most exciting sort of endeavor in the lab that is also finding a lot of interest going forward.
Yeah,
So,
fascinating.
yeah.
So most drugs that can be delivered intramuscular can be delivered through the microneedles.
Correct. Yeah, a lot of those drugs can be
Interesting.
delivered. Yeah, um, transdermally. One of the limitations of the transdermal delivery is the quantity of the drug you can deliver. So you can take a lot of tablets, for example, or you can, give a large quantity of drugs through an injection. But microneedles limitation was always the quantity of drugs. So if you're looking at high potency drugs where you don't need a large quantity, but of drug, like if you just want to build up your immune system with a vaccine, or you're delivering very small quantities of chemotherapeutic drugs or some immunotherapy drugs, then microneedles are ideal for that, because you don't need a lot, and all you need to do is activate your immune system or deliver a small amount, and that can be done transdermally really well. And so we had developed technologies to streamline the process of manufacturing of those microneedles so that we can make large quantities of this. The microneedles had been around before we started working on it, but our technology makes it more scalable and and so forth. And there are many applications for microneedles beyond just vaccines. So it's a platform technology like the pill. So you can use it for many different possibilities. And I think there is a promise there for that. Uh,
Yeah,
yeah.
that's a whole other podcast, but I could have used that when I was 5 years old. Every time I went into the doctor and was sick, I got a shot of something, probably
Oh,
antibiotics.
yeah, yeah, yeah.
They used them too much back then. So, and that was my greatest fear
Yeah.
is getting a
Injections,
shot.
right? Getting shots,
Yep.
right? So this will take that fear out. I think 30%
Hmm.
of the population is afraid of taking a shot, right? They have this needle phobia. And so that's one of the things these microneedle patches also address, because you don't see the needle. You just put it's like a nicotine patch or a patch that you just put on the skin. So and it does have that advantage compared to other methods.
Yeah, I can see why it's catching on. You mentioned the ingestible microbiome sampling pill. I forget how I stumbled upon this, and it fascinated me as soon as I heard about it. And for our listeners' sake, it's a complex uh, invention, but also I think relatively simple in its essence. You just ingest a pill, which then samples all throughout the human digestive tract, then can be sequenced similar to how fecal samples are currently sequenced from the colon. So just curious how this was first conceived.
Very great question. And that was a great explanation of how the pill works, right? So you take the pill and it goes into your GI tract and it acquires a sample. from where you wanted to acquire the sample. So it's like doing sample collection from inside the body, right? Which was, and in medicine or in healthcare, I think our GI tract, I call it like a black hole. Like people don't know much about what's going on in there. Uh, the biopsy probes, you can't put it every day. You have to be really sick to go to a doctor and sort of investigate what's wrong. So, and it does not really access, for example, the small intestine, which is you can access from the top all the way down there, and you can get to the colon from the bottom, but in the middle there is this huge area, and that's very significant. So what was happening was, the reason why I was not working in this field many years ago. I had actually approached a program manager who was managing a large program for microbiome researchers. And I had some technology at that time to sort of investigate. I was not doing microbiome, to investigate our GI tract. This program manager basically indicated that the whole field of microbiome is advancing fairly rapidly, but everyone's been sort of analyzing feces. They were using fecal matter to derive their conclusions as to what's wrong, how microbiome influences health, and so on. But that gave a completely incomplete picture, because there was no way of really knowing which microbes were in which part of the GI tract and how they were really influencing the host, right? So that information was always missing. In spite of that, a lot of progress was being made in microbiome sort of research, and it's still being made by just analyzing feces, but they cannot draw the link. They know the correlation, but the causality of how microbes affect our health or how we affect the presence of microbiome, that information, that link is actually not very well established. So this program manager indicated that we want technologists or engineers like you who've been developing all these technologies to think about how do we help these microbiome researchers to get that information. And so once you posit a question to an engineer, that's the best thing that can happen to that person. Engineers can solve problems, but you got to give them the right question to answer. And the moment that question came in front, I mean, it was a challenge. And, like most engineers, we would like to wear that hat and see if we can come up with a solution. And that got me started into the field of microbiome to figure out what challenges there are and how we can make that happen. So it's a long story, but I think it does start with defining the problem first. Somebody has to identify a gap in what is in the understanding, and then saying, you know what, this is what is missing. We need something to fill this knowledge gap. What can you do? And that's when, if you have the right question in front of you, and if you have the training or the background to be able to do that, then you can think about solutions.
Yeah, it makes sense. Was in all of your research and background, was this your first venture into the microbiome, or have you done that
Yes.
before? Yes.
No, this
Okay.
was my very first venture into the field of microbiome. Trying
Yeah.
to,
Wonderful.
yeah, thank you.
Yeah, and you said obviously most of the small intestine is not accessible. Needle biopsy, is that pretty much the only way you would access it?
Yeah, I mean, people do try sort of needle or surgically open up, or that would be one of the ways. That is a very invasive way of getting access to the small intestinal microbiome or information in the small intestine. Correct. Yeah.
Yeah, I can see why you call it a black hole. Yeah, that makes sense. It makes me wonder how what they're drawing upon currently. You talk about SIBO, small intestine bacterial overgrowth, and how do
Correct.
they even know what's in there? And I think that's part of what you're saying, right? We're
Correct.
making conjectures, but we really don't have much information.
You're right. I mean, they say that IBS or Crohn's or colitis, the small intestinal bacterial overgrowth, as you just mentioned, is sort of a way to diagnose that. But and that is just a conjecture until you can really prove that to be true. And how would you prove it unless you really have access to it, to that, to the small intestine? So I think this ingestible sampling device finally allows you to collect sample from the small intestine, and hopefully allows scientists or researchers to figure out that dependence or that link between different diseases, and in this case, small intestinal bacterial overgrowth, or any dysbiosis in the diversity of the microbiome population in the small intestine, or maybe a colonization of a certain dominant species that shouldn't be there in the small intestine. And all that information it will only be possible if you can get local access or information in the small intestine. Yeah.
Yeah, it's sort of, this is maybe an odd analogy, but it occurred to me sort of like building a rocket to the moon. It's like you don't really care what goes on the moon, but if you need to get somebody there, you need to develop a rocket ship to do that. So you've developed an internal ship. Really, it really is sort of a ship traveling all through the
You're right.
digestive
Yeah.
tract.
That's a
So.
great analogy. Yeah, you're building a rocket ship, and then you can explore the frontiers of space. That's a perfect analogy. This is the frontiers of the gut. You can explore the gut frontier using this new pill device.
And you can let the microbiome guys largely do that, but you're helping them in a way that is really impossible. That's just fascinating.
Exactly, exactly. That's basically how, yeah, it's like how microscopes are used to study cells biology, to identify the structures of cells and tissues. It's like a tool, it's like an instrument, it's like a method that will allow you to now study in much more detail what's happening inside the small intestine, which was not possible before because you're only analyzing feces, and that does not provide a complete picture of the gut microbiome.
Yeah. As I'm sure you know, the research on the microbiome is just growing exponentially. And once this takes off, won't be able to keep track of all the data. So that's
Correct.
very
Yeah.
exciting.
Yeah.
Yeah. So this pill is standard size pill. It's 3D printed. So you can help us understand what it looks like and how it actually is grabbing those samples, and how many samples can it get, and can you specify where they come from, that sort of thing.
So yeah, that's a good question. So we have many different versions of the pills that we've been working on. And one of the pills that we published, and one that has been approved for upcoming human trials when we get there, is this a soft elastic pill. It's essentially passive. There is nothing in it. What we found out was that our peristaltic pressure that the gastrointestinal tract has is able to push the sample that we want to acquire into the pill. So it's a very simple device. It has inlets on two sides. We rely on external GI tract to actually push the sample in. We don't have to make a pump. In our prior version, which I can talk about as well, we actually put a pump in there to get the sample, but we realized that we probably may not need that because the body does sort of push the sample into the pill. But the key is once you acquire the sample, you have to lock that that sample that you acquired. Otherwise, it'll get contaminated because you're probably only interested in sampling from a given area of the GI tract, right? So once you collect the sample from there, if the inlets are still open, then you would basically get contaminated as the pill passes through the GI tract and then comes out on the other end. So we have a mechanism inside that actually has a valve. Once the pill is full, it automatically locks itself or closes itself from the inside. So it's like a self-locking mechanism. Once you fill up the pill, it will lock itself, and then it will not let any more sample in or sample out. So that way you have the sample is preserved. You collect the sample when it comes out on the other end in the feces, and then you can analyze for whatever you want to study about the small intestine or wherever that pill sampled from. So the way it decides where to sample from is based on the enteric coating the pill has. The pill has a very specific enteric coating, and it dissolves based on the acidity in the GI tract. So for the listeners who don't know, the acidity is different in different regions of the GI tract. So in small intestine, it's slightly higher than the stomach, and then the colon, it's different. So if you can tailor the composition of this enteric coating, it will only dissolve in a targeted region of the GI tract. And once it dissolves, that's when the sample can get into the inlet. So you control the start of the sampling process through this enteric coating that you have on the outside. So that's how it's a very simple device. It's a passive device. It has no batteries, no electronics, so it's very safe. You could, it's essentially as safe as taking any other pill that you would take. And it can be made in many different sizes. At the moment, they are the size of a small vitamin pill that you can take, and it can be made larger or smaller based on how much volume of sample you want to collect.
So without that enteric coating, it might only grab samples in the first few inches of the small intestine.
Correct. It will just
Yeah.
basically grab, if you don't have any coating, it will probably fill up in the stomach itself, right? So, so you at least have to protect you have to have a coating to protect the pill in the stomach because you want to sample in the intestine. So you have to wait until it gets into the small intestine. So if you do not have this enteric coating, it will just acquire anything that fills up. So you sort of delay the sampling process by controlling the composition. You also tune the composition to decide which pH. And in the future, you can also tune the composition based on some other marker in the GI tract, right? So there's a lot of possibility as to how you make the enteric coating for sampling.
Do you know, like, say somebody has SIBO, depending on the pH of those pathogens, could that sort of throw off the sampling? You know, maybe it raises the pH. The pathogens tend to be of a higher pH environment. Is that a
Correct.
possibility, or?
That's a good question, Richard. So you can tailor this. It's very well known that the acidity would change in the environment with people who have small intestinal or bacterial overgrowth, and that is precisely this will also allow you to detect, right? So if you made the enteric coating such that it dissolves at a given pH, and if you see a lot of sample being collected and it has a large amount of bacteria in it, you would then be able to say, yes, this person does have SIBO, and these are the bacteria that are actually influencing it. So, and let's say you didn't have SIBO and the pill entry coating did dissolve and you collected the sample and there is nothing in there or the bacterial diversity is very low or the quantity is very low, then you know that there is not enough SIBO. So you can actually use the pill to sort of diagnose SIBO in the future, and also study the bacteria that are populating. And it's a range of pH, so it's not as if it's not going to work. So you can tailor the range of pH as to where it would sample from. So you know you're getting the sample from the small intestine.
Yeah,
So
interesting.
yeah.
And did I hear you correctly? Are there 2 sampling sites or more than 2?
Each pill will only sample from, at the moment, from one area of the GI tract. So if you want to
Oh,
sample
okay.
from multiple areas, you take multiple pills. So for example, let's say and it's as easy as taking a pill. So that's why we find this a much more simpler and elegant solution. And each pill could be targeted based on their enteric coating to target the different region of the GI tract. And so when the pill comes out, you can have one that is in the colon and in one that is coming from the small intestine, for example. Or you could look at in the distal end of the small intestine or the upstream of the colon. So you can decide where the sample is coming from. And you can collect all of those samples by using different pills.
Yeah, that makes sense. So do you do any of the testing there, or do you send it out to another lab? And what is the method of sequencing that's currently being used?
Yeah, so we have a very strong collaboration with the Tufts School of Veterinary Medicine, which is out in Grafton. I'm very close to Boston, and Grafton is Western Massachusetts. And that collaboration, we have been able to validate all these pills in different animal models. So we tested them in pigs as a very good animal model. We also tested them in dogs. So these are hounds, many different dog species. We tested them in dogs as well as to figure out how well the pill sample. So it has been preclinically tested right now. Uh, it hasn't been tested in humans yet. So so we have the data that actually now allows us to translate to humans. So we'll be doing some of our earliest, first studies, pilot studies in humans, hopefully in the coming year. So for analysis, we perform very standard 16S ribosomal RNA sequencing, which is the standard for identifying the bacteria and to identify the diversity of the bacteria. And we do high throughput sequencing. We do have a lab ourselves. We do have a lab at the Grafton campus where we analyze all the data ourselves and basically try to identify the bacteria and the taxa. And that's how we've been able to confirm the. So we find that to be much better than culture methods, of course, because that takes a lot of time.
Yeah, for sure. Yeah, 16S seems to be the most common, with shotgun metagenomics growing a lot. So in theory, once this hits the consumer market, the consumer could take that pill and send it to any lab they wanted.
Yeah. So at the moment, the business model is not fixed. We don't know how people want to use it. And you brought up a business model right now where people could take their own power into their own hands. They analyze, they take the pill, they collect the sample, and they send it to the lab of the choice where they can then get a report back to identify what bacterial species inhabit. And they can then sort of correlate based on what people know from the literature, what's wrong with them, right? So there are multiple use cases for this pill. It could be like the 23andMe kind of a gene sequencing. Here, instead of doing that, you're doing microbiome analysis of your GI tract and trying to find out what's happening with your gastrointestinal system, right? So there are many possibilities here for how the pill can be used. Yeah.
Sure. I mostly use 16S tests. I've done one shotgun, and I appreciate it can go down not only to the subspecies, but the strain level, which is
Right.
fascinating, can be helpful. But I can imagine, especially in the early days of gathering all of this data, that just is probably too specific. Sounds like 16S will be more than enough, at least
Yeah,
initially.
yeah. I mean, I think the more data, the better. So now you have a way of collecting data from a region of the GI tract. You can repeat the same test that you've been doing on the feces, but now on a different sample. And as we gather more data, we will learn more about each of the diseases and what's happening with you and your relationship with your microbiome. So I think, but at the moment, since we are at the very early stages of what can we do with the pill, I think 16S definitely is the easiest analysis to perform to find out the bacterial information in your GI tract. Yeah.
Yeah, makes sense. Since this is not a drug, you're not bound to those sorts of regulations, but what regulations are you have to go through, what hoops to go from research to commercial production, and how is that going?
So this is a good question. I am not an expert on the FDA regulatory aspects of things. I do understand some of it. As you rightly pointed out, it's not delivering a drug. It's neither detecting a disease nor is it delivering a drug. So, and it is a pill that does not. It basically goes in and it comes out on the other end, right? As it goes in. So the and it's right now we do have an IRB approval, which is a human pilot study approval. We were able to get that under a non-significant risk designation, so NSR designation. So it's considered a non-significant risk device. So I am hoping or expecting that the regulatory pathway for this pill would be better than any other implantable or ingestible device, which uses that device to diagnose or deliver drugs. We are not doing either of them. We are just basically collecting a sample. And so as long as we can show safety in the humans, which we have shown in animals in the preclinical model, and our upcoming studies would do that, I'm hoping that we should be able to get we don't know exactly which pathway, regulatory pathway it can take, but it shouldn't be as complicated as some of the other devices or drugs in the market.
Might you have a beta testing phase that would be open to anybody that wants to volunteer for that? I'm part of a 14,000 member Facebook gut club group,
Oh,
and they
really?
talk about SIBO all the time. And I could think a lot of them would love to be part of the beta testing. Is that a possibility?
So we do have a study coming up. If it gets at that stage, it is fingers crossed it might happen, and there will be we will be recruiting some pilot volunteers, but this is just for diet study for a very specific case. As soon as we are done with that, definitely I think there will be an opportunity to for people to volunteer for a follow-on study when we go to the clinical trial phase of this pill, when we are trying to at that stage, we'll be trying to assess how well the pill samples from the GI tract, because we want to validate the pill first. But the opportunity there is then it will also be able to collect the sample and then analyze and understand the differences. But I think the first step is to validate the safety, assess the safety of the pill, and to ensure that the pill is indeed sampling as expected from the targeted region of the GI tract that we say it would. So I think the initial studies in humans would basically be focused on that.
Other than a simple Band-Aid, it seems like the safest medical device you could possibly use.
Yeah,
So hopefully
exactly.
that'll go rapidly for
Yeah,
you.
there's
Yeah.
nothing in there in the pill, as you point, it's not delivering anything, and it's not even telling, it's not diagnosing anything either. It's just collecting sample. I like to say it's a better fecal collection device, right? Instead of collecting feces from the outside, you're just basically collecting feces from the inside, if you want to think it that way. So it should be, hopefully it should be easier to get through the regulatory approval. Yeah.
Sure. Well, I know there, as I implied, countless people who'd like to get on that rocket ship you're building and get some more data.
Yeah,
So
I'll definitely
that's
reach out to you when I'm at that stage, to reach out to your gut club to tell you about the progress and how they can sign up. Or at that point, when we reach that stage, I'll be very happy to do that.
that would be an honor to be part of that. Yeah. So and I realize, this progress to market, it's really hard to identify, but so do you say you've actually started the human testing or about to do
So
some of that?
we are about to start human
About to
pilot
start.
study. About to start, and we still have to clear one level of funding approval before we get there. Uh, Fingers crossed, as I said, we will get that, and then we will basically begin the process this year. Um, you know, uh, and I think we are all ready. We have the approval. The IRB approval is the longest process. It takes most time, and since we have that approval in hand, we only have to get the funding and then start recruiting volunteers from our database and to do the pilot study.
So you think 3 to 5 years before commercial to the public?
I'm more optimistic, so I'm saying,
Ah,
yeah,
great.
I'm thinking less than 3 years. If the pill works, and if all we are doing is sampling, that should come out fairly soon, right after we get pilot studies. We should be able to put the package together to the FDA and get that going as soon as possible. Yeah.
Yeah. Like you're implying, it's um, what do you need to prove? Nobody died. It didn't hurt anybody. It does what it says.
Correct.
So that makes a lot of sense. Yeah. Maybe an impossible question at this stage, but any wild guesses as to, and again, you said it depends on the business model if you're doing the testing as well, but how much something like this might cost?
So a business person would say the pricing depends on demand and supply. So, and a scientist like me would say, how much did it cost to make the pill, right? And so the pill is and that price is very low. So to make the pill, we literally use very standard materials that are biologically inert. It's used already in lots of surgical devices and tools. These are just polymers and plastics, 3D printed or injection molded. It doesn't have to be 3D printed. It's a very simple device. So the price to make the pill is below $1. It's not going to be that high. But again, the supply and demand, the quality control, the and what market can pay, and the analysis that the business folks do is very different than what scientists sort of look at. So I wouldn't if I were to if I had the authority to price it for general use, I would say that $1 a pill would probably be a good guess at the moment, just for in terms of making the pill and sort of doing it. I haven't added the cost of marketing or business in there, but I mean, I think that would be sufficient to make the pill and use it. The the analysis of the pill is more expensive. So when you send the sample to a company and then you get it analyzed, that would cost you.
Of course.
So yeah, and so the pill itself would not be as expensive as what the follow-on analysis would cost you.
Yeah, that's a great answer. Of course, we won't hold you to any of those costs, obviously, but it's very encouraging because I assume this might be a difficult reach for an average consumer. And what I hear you saying, at the very least, the sampling part won't be ridiculously expensive. Yeah,
Won't be ridiculously expensive. No. Yeah.
yeah, that's really good to hear. I'm sure our listeners would be glad to hear that. Um, Fascinating project. Is there anything that we haven't covered that would be useful for our listeners to know about this project?
So I think what I would like to say is, which I started with by saying that the small intestine plays a very important role in health and disease. You brought small intestinal bacterial overgrowth, but the small intestine is not just playing a role in digestive health, because it is it's an important part where the microbes synthesize vitamins, it facilitates absorption of nutrients, but it also is very responsible for the immune function, right? So because a significant portion of the body's immune system actually resides in the small intestine, and that's why we have IBD, inflammatory conditions, and autoimmune disorders. So the reason I'm mentioning this is because while the pill is just the beginning, we want to know more about that region, how it affects digestive disorders, but also autoimmune conditions. The pills can get much more interesting as we progress the science and engineering. So right now you can have pills that are sampling the microbiome, but you can also sample the metabolome. You can analyze different aspects of your GI tract. You can also have a pill that would actually be much more targeted in the future. You don't want to sample from a region, but you want to be very specific from, okay, I just want to get a sample from here. So a next version of the pill could be a more targeted pill, right? So this is not the end. What I'm trying to say is there are lots of opportunities and open questions and very interesting things to develop technologically on my end, but also interestingly to explore from the medicine and healthcare point of view. And I think there are interesting possibilities for I call it a lab on a pill device. Ingestible device that samples is one, but one where you can do sample collection, processing, some sort of intelligence in the pill would actually be the next frontier for some of these ingestible devices. And I think there is a lot of activity in this field now. After having seen what's happening in ingestible devices, I see what I have been doing, but also a lot of other researchers have been exploring many different ideas for what can go into this ingestible devices. And I think there's a lot of interesting things that's going to happen in the coming years. So it should be exciting.
Yeah, certainly changing healthcare as we've known it. And I think of like the continuous glucose monitor, which is pretty common, but
Correct.
would you see one day something like a CGM that is able to get all sorts of, like you said, real-time data and send it to a phone or device or whatever?
Yeah, I would love to see that microbiome sampling as we learn more and how significant microbiome is to our health and disease. maybe a time will come when we go to a doctor and the doctor prescribes not just a blood test for your annual physical, but suggests you do a microbiome sample using a pill, find out what's going on. So you're not just collecting blood panel, you're controlling, you're getting a microbiome panel for your health. And when anything is wrong and with you, that this becomes as routine as taking a blood sample for diagnosis and for managing of your medical condition, to see how well you're responding to treatment or what's going on with you. I think there's a lot of information there that can be had from this field. So that's the hope in the future. It could be as routine as a blood test.
That would be encouraging, because my experience and most people's experience, I think, doctors don't even know to test the microbiome yet, let alone use a pill. So, that's not your problem, but it's a major healthcare problem that I think needs to be solved.
Correct. You're right.
Yeah. Well, last question. And again, so appreciate the time you've given us. This is the Resilient Biome podcast. So wondering what your top recommendation might be for our listeners that they could do to have a more resilient biome.
So that's a very good question. I am not a clinician or a healthcare practitioner myself, but I have studied the role of diet in microbiome, and our upcoming human study is going to be investigating how small intestinal microbiome varies with diet and nutrition. And so the nutrition and diet space is personally very interesting to me. And so what I would say is, from what I have read and what I understand, talking to GI physicians and nutritionists, is that taking highly processed foods or excessive sugars or diets that are only one-dimensional affects your microbiome in a negative way. So having a much more diverse diet, staying away from processed foods, and not taking a lot of things that would insult the microbiome, like antibiotics or repeated use of antibiotics or alcohol use or any of those that have a strong microbiome insult that affects the microbiome. To maintain the diversity of the microbes, having a diverse diet and the balanced nutrition would probably be very helpful. And I think the pill, as we study in our human studies, I mean, I'm sure we'll probably learn more about how nutrition affects the small intestinal microbiome, and we'll be able to understand how that happens. But at the moment, I think it is commonly understood that avoiding processed food and avoiding microbiome insult, taking antibiotics or things like that is probably a good thing to have a more resilient and a more stronger microbiome in the small intestine. Yeah.
Yeah, that's a tremendous answer. I don't think folks realize you're always feeding your microbiome. You're either feeding the bad guys or you're feeding the good guys.
Correct.
So diet is absolutely key. Yeah, thank you for
You
that
got that
answer.
right. Yeah.
Yeah. Well, again, thank you for your time. This has been fascinating. I'm going to keep an eye out for new press releases, and I just wish you the best with this and all of your projects.
Thank you, Rich. Thank you for having me. This was a great conversation. I appreciate the opportunity. Thank you.